distinguished colleagues and friends, I would like to welcome you on behalf of the Gerontological Department of the Russian Academy of Medical Sciences. I see so many colleagues and comrades here. I'm sure this conference will be of interest to many of you, to guests and to speakers. I'd like to thank the organizing committee, the program committee of the conference, and in the previous speech, which was made by Mikhail Batin, a very interesting idea was put forward. The aging and disease were considered similar phenomena. That's why I'm going to speak about medicine against aging. Do we have it or not? And I'll try to tell you what science can answer to this question. Here you can see that the main diseases, age-related diseases, are increasing and aggravating in the process of aging. Everybody would like to live till 100 years without any handicapping diseases, but how can we do that? That's a good question. It's a tough question indeed. Here you can see, for instance, that if we discard the most handicapping diseases, our lifespan wouldn't increase that much. You see, by only 2 years we can increase our lifespan if we eliminate diabetes, cancer, and so on. So you see, only eliminating diseases would not solve the problem of longevity. Here you see the main groups of geroprotectors, the most famous ones: antioxidants, some neurotropic drugs, hormones, peptides, anti-diabetic drugs, immune-modulating drugs, and others. It's a long list of all possible groups of gerontoprotectors, but I'd like to focus on the main groups of them, which can already be used to prolong our healthy lifespan. And here I would like to show you the list of drugs that are used by the National Intervention into Aging Program of the United States. You see, these are NSAIDs, some antioxidants, and some even a little bit unexpected drugs I can see here. And by the way, here you can see the list of the pharmaceuticals and substances that were tested in our laboratory of the Research Institute of Oncology, Laboratory of Carcinogenesis and Aging. And you also see neurotropic drugs, peptide bioregulators, micro agents, enterosorbents, and rapamycin. And in this audience, I guess everybody knows that one of the most popular theories of aging is the free radical theory of aging, which was suggested in 1956 by Dan Harmon. I hope he lives till 100 years and will live long enough to receive his Nobel Prize he's been nominated for. And you see that gene grafting can inactivate free radicals and can prolong our lifespan. And we saw this in Drosophilus, but not in mice and rats. Here you can see some of the effects related to antioxidants. The majority of them increases the average lifespan. Some of them stimulate fertility, some of them decelerate it. But the problem is that some antioxidants can turn out to be carcinogenic. That's the problem, because sometimes you cannot be sure of safety of drugs if the patient has to take them for a long time. Talking about antioxidants, multi-center epidemiologic study was made, and it was proved that alpha-tocopherol, or E vitamin, provided no more protection against heart attack or stroke in high-risk patients than it did in a placebo group or in control group. Beta-carotene can, in high doses, stimulate cancer, lung cancer in smokers, and we don't have any proof that it's the geroprotector. Academician Kulachov, working at our institute, he suggested using Kulachov ion. He binds free radicals in mitochondria. So these are mitochondria-oriented antioxidants which prevent free radicals from getting into cytoplasm outside mitochondria. In many centers of the world, these studies were carried out, and it was proven that it prolongs the average lifespan, but it does not in any way protect against development of cancer. And again, we don't see a maximum and definite increase of the maximum lifespan. Here you see data on resveratrol, which was a big trend. It was suggested that red wine was the salvation for us. And we saw that mice who were taking resveratrol and high-lipid diet, it increased their lifespan. But in humans, to achieve the same effect, the humans would need to drink 10 bottles of red wine per day for longevity. I'm not sure even the heaviest drinkers would be able to drink that much to live longer. Of course, we also understand the importance of nutrition in the project of aging. Here you see the details of the experiment which was made on rhesus monkeys, on squirrel monkeys. Since 1987, an experiment was made which consisted of reduction of the calories they consume per day. And you see the latest results published last year by Chemnitz, and you see the incidence of cancer is decreased, the cardiovascular diseases incidence, diabetes is decreased as well. Not long ago, another publication was said that this does not affect in any way the length of telomeres. But if you take light food, you will have higher sensitivity to insulin and higher tolerance to carbohydrates. And the studies were made on nematodes, on monkeys, on rats and mice. The life of them was increased in all the cases. Then for 6 years, a study was made on humans. They consumed not more than 2,000 kilocalories per day. They restricted the sweets they intook, and you see 18 persons in control group, 18 persons in calorie restriction group. And the total result was hard to assess, by the way. But what was proven for sure, that atherosclerosis development was much prevented by intake of smaller number of calories. But the society has actually proven it to itself, because if you compare the average daily consumption of nutrients, you see the highest figure is in the United States, and you see the large number of obese people in the United States. In Europe, it's 17% less. In Japan, it's 27% less than in the United States. And you see how it correlates with the lifespan of males and females. In Okinawa, it's 50% less than in the United States. I mean, the calories consumption, but these people, they are small, they have very low body weight, and they have the maximum number of centenarians. Of course, we understand that in Canada, in the cold climates, you wouldn't be able to survive unless you take a lot of calories. Of course, all of us would like to live long without restricting the calorific value of our nutrition. And talking about anti-diabetic drugs, Vladimir Dilman in 1970s, he worked in St. Petersburg Institute of Oncology. He suggested using biguanines, buformin, fenformin, and metformin. Here you can see the main effects resulting in increased lifespan, and that can explain how anti-diabetic biguanines can increase the lifespan. And first of all, susceptibility to insulin is underscored in results of all these studies. 8 years ago, Spindler from USA proved that the gene expression changes that are common to metformin and CR, they were associated with the following mechanisms of aging. And here you can see the results of our study we made together with Vladimir Dilman in 1980. And it showed that biguanine can prolong the lifespan by 24–25% and can dramatically reduce the cancer incidence in mice. Here we use transgenic mice with this cancer gene, which triggers up to 40% of breast cancers in humans, while metformin decelerated the development of cancers and prolonged the lifespan. We used outbred mice, ASHR line mice. It's a Swiss line of mice, and the increase of their lifespan was up to 28%, and the maximum lifespan was increased. And what's important, reproductive function was prolonged. It wasn't inhibited, it was prolonged. And you see that senescence, the number of senescent cells was reduced. This is one of the major results of this study. And here you can see results of in vitro study of metformin impact on breast cancer. It was proven that metformin inhibits expression of cyclin D1, HER2, and others, those who do trigger development of breast cancer, if not inhibited. Here you can see results of an important experiment in which we try to answer the question, at what age should we start to take geroprotectors? Because some people think, okay, when old age comes, we'll start taking them. I think that after the sexual maturation, we should start thinking about prolonging our life. Here you see that metformin, if you start taking it from young age, it will prolong your life. But it depends on at what age you start taking it. This is an unpublished result of a study, a very interesting one. Metformin was injected in newborn male mice, and you see that the earlier you start taking these geroprotectors, the longer you can live. Interesting results, really. And here you can see results of another study It's a study on mice using 1-butylbiguanide and how it increases lifespan again. Here you see how buformin and fenformin in weight studies, in old rat experiments, again impacted the rate of aging. Here it was proved that spontaneous cancerogenesis was inhibited by buformin in female rats of Lyon line. This was published 30 years ago. In this result, you see that X-ray radiation, which is a very effective thing, it shows that fenformin also inhibits carcinogenesis. Different models here prove that anti-diabetic biguanins can prevent development of cancer induced by various carcinogens. Here you see, for instance, the effect of metformin on development of skin cancer, of melanoma. And here you can see the inhibitory effect of anti-diabetic biguanides on memory carcinogenesis in rodents. You see all the different carcinogens and their effects, and how biguanins can decelerate this process. Or for instance, metformin prevents development of cancer in colon of mammalians. And here, not only in colon, not only breast cancer is its development is decelerated, it's also true to the cervical cancer and other types of cancer. Here we summed up the data on animals who have been used in experiments till now, and you see a large number of publications. More than 40 reviews and meta-analyses were made over the past 2 years in PubMed, proving that metformin can decelerate the development of cancer or prevent development of cancer. Here is an interesting scheme which shows that signaling from insulin, glucose, the signaling can be inhibited by hormone turning off or hormone receptor turning on, reduction of calorific value of nutrition or caloric restriction. All of these things influence on signaling. Metformin affects all these weak points. This is what Mikhail Bogoslovny is going to talk about in the coming days. He shows that how metformin influences mTOR, which is a protein kinase regulating the cell proliferation, because cell proliferation is the key to aging and to carcinogenesis. Here I try to sum up the data on the influence of various methods of increasing our lifespan in the systems of insulin transportation to the tissues, for instance. And you see that hormone knockout mice, in mice you can increase the effect by 50%, but they usually restrict fertility. Biguanides, they stimulate fertility. This is one of the best regulators for fertility. It was shown in 1979 in our laboratory. And you can see here that the number of publications on the impact of biguanines on cancer is increasing dramatically since the beginning of this century. And even in Rejuvenation Research magazine, it was published that metformin is a geroprotector in animals. Now we have to prove this in humans. But in Science, in January this year, a comment was published saying that metformin reduces by up to 40% the risk of cancer of any localization. So this seems to be the most effective means in the whole history of cancer that has saved a lot of people from cancer. The next stage was studying rapamycin. Another publication was made by Harrison, Strong, Sharp, and others saying that the hybrid mice lifespan can be increased this way even if you start taking rapamycin at an older age, and if 9 months old mice start taking it, this too will increase the lifespan. But the problem is that in these publications I didn't see any indication of the cause of death of these animals, which would be of interest to see. Here you see, for instance, that using rapamycin increases the lifespan and inhibits spontaneous tumorigenesis in inbred mice. Here you can see some data such as rapamycin effect is not very strong, but it does decelerate the development or prevent development of cancer. 129 SV mice, you see here uterine tumors of different genesis. It's hemangiosarcomas, carcinomas, and so on. And this is another publication we made in 2010 in the American Journal of Pathology, showing that rapamycin inhibits the development of breast cancer in cancer-prone mice and prolongs their lifespan this way. It was already mentioned that there might be an impact of the environment on the process of aging. I'd like to take a special look at the day and night induced mechanisms. You see that turning off the night peak of melatonin, which happens somewhere between midnight and 3 a.m., it results in disruption of circadian rhythms. And if you turn on the light, or if people live in the north, where the polar night is when you don't see the sun, but the sky is blue, it's like in St. Petersburg, all this results in higher tumorigenesis risk and inhibits melatonin. When we analyze the data of the International Agency for Cancer Studies, and we see that in this case the cervical cancer incidence is higher, breast cancer incidence is higher, and it depends on the continents. Of course, you know that stomach cancer is associated with Helicobacter pylori and other problems, but the question is, what should we do in this case? Not long ago, a work was published in which there was a question: why did the breast cancer incidence in Alaska increased over the past few decades by nearly 30%? Threefold, sorry. Probably this is because of development of this area after the oil and gas reserves were discovered. And you know that at night the lights are on in polar cities, like in big cities. You see here that we switched off the gene, the TAU gene, and if we switched it off, it 30 to 40%, and if we which means that circadian rhythms are very important to maintain, to enhance our longevity. Now up to 29% of all people work at night. It's not just doctors, policemen, but also common internet users and computer users. They like surfing on the web at night instead of going to bed. All this results in the increased incidence and risk of metabolic syndromes, cardiovascular diseases, and other age-related diseases. Here you see that mutations in circadian genes produce such late effects as lifespan reduction, tumorigenicity, and others. And here, for instance, you can see where people with different malignancies have disrupted circadian rhythms. This work was first published in 2008 in Israel. Now we have data for 168 countries, and if they have such a life, you see how Earth is lighted at night, and you see in the areas that are light blue, these areas have the highest incidence of cancers, of tumors. Again, night insomnia increases the cancer risk in females. This is especially true for females, women who work in shifts, who work the night shifts. Of course, there are a lot of various factors. It's nutrition, diet, environment, family, genetics. Effect of light regimen is easy to test in female rats. And we see how carcinogenesis is triggered. And if we give rats not the standard quantity of light, 12 hours a day, but if we give them less light, we see that they feel better. So light at night promotes, and darkness inhibits carcinogenesis and mammalians. And this was very well correlating with melatonin and prolactin levels. This correlates also with the colon. If animals live all the time with light, the rate of carcinogenesis will be higher. If they live a whole day in darkness, the tumorigenesis is much reduced and they live longer. Look here at these mice of this HR2 New Life. We see that injection of melatonin can inhibit the expression of this cancer gene. With age, these mice, like everybody else, have a higher incidence of various tumors and problems. And by the way, here are the results of the experiment where rats were kept in in light and darkness in Petrozavodsk. In Petrozavodsk there is a white night. So they generated various peptides, and here you can see that when the animals are used in the natural light, they are better. You see that rats and use of melatonin or synthetic peptide can prevent the development of these pathologies, reduce the carcinogenesis, and increase the lifespan. So effects of light-dark regimen on lifespan and spontaneous tumor genesis in female rats is shown in this slide, and you can compare pre and post injections results. When we, together with our American colleagues at Baltimore, looked at the expression of genes in these animals, they were receiving melatonin, and it turned out that it all influenced the genes that do participate in the aging pathogenesis. And here, for instance, you can see a few results. You see the skin tumors. They are far from endocrine system, but again you see the effect of melatonin on these systems as well. So we have the right to say that at night we age quicker. At night we age much less than in light. Here you see different models of using melatonin, but our institute has already made new studies, some of them on humans. And for instance, for breast cancer, colon cancer, and other cancers, it is a solution because we see that the lifespan really increases. This is a conclusion of the International Agency of Evaluation of Carcinogenic Risks on Humans. And here you can see results on another very important event. More than 30 years, we collaborate with Professor Havinson in St. Petersburg. His laboratory has created this whole number of peptide drugs, and we studied their impact on carcinogenesis and lifespan with Drosophilus, with mice, with rats. In all the experiments, their lifespan increased by 20 to 30%. And here you can see, using various carcinogenesis models, how these drugs prevent the development of cancer. And you see there is no complication. It was also showed that used and you see how you can restore the level of night melatonin. And in humans, the same thing happens. Melatonin level, in case of taking another gram, can be reduced. And these are the results that were obtained by academician Kompushka in Kiev, and they proved that death rate is much lower if you take epiphysis and thymus. And this study was made in a special home for old people. They lived quite comfortably, and their lifespan was much longer, which is an achievement in this area, I think. The lifestyle effects. And here the information is of real interest. It makes us think whether we need to take any pills or not. Because it turned out that men with high intellect, they live much longer than men being rather dumb and stupid. But we saw a reverse effect in women. Women live less with high IQ than women with low IQ. So the idea is that women with higher IQ live with more stress because they have to prove to men that they have a high IQ. But the longer you study, the longer you live. That's interesting. The Nobel Prize winners, they live longer. Especially Nobel Prize winners in medicine. Probably they know the secret and keep silent about it. Because economists too live longer. It was also proved that the members of Russian Academy of Sciences live longer than corresponding members or doctors of medicine, medical sciences. The second part of our lifestyle is physical activities, exercise. And it turned out that athletes, members of national teams of each country, those who were involved in aerobic types of sports, unlike those who were heavy lifters or athletes involved in some heavy and static sports, they lived longer, the ones who were involved in more active sports. You see that mortality rate in elite athletes, how it looks like. Same for footballers and hockey players. They live 4 years longer, while those who do the weightlifting, boxing, wrestling, they live longer but not much. So the question is, do we have a choice? Here you see all the genetic modifications that are used to prolong animals' life. In mammals, it also modifies to this or that extent development of cancer. If we inhibit aging, we increase the risk of development of cancer. Deceleration of aging reduces the risk of cancer. Look at this picture by Vasnetsov. This warrior is standing in the field and thinking which way to go further. But we know that our cells age and die. If carcinogenic factor influences the process, the cell will turn into a cancer cell. And a latent cell, in case of promotion, is transformed and cancer develops. Here you see some genes that influence the process, and you see these same genes influence the aging process and development of spontaneous tumors. And this balance of this process can result in very unpredictable consequences sometimes. Calorie restriction. Some of them I've been talking about. It's melatonin, big one needs antioxidants and so on. They mimic the effect of restricting the calories diet. And again you see the carcinogenesis mechanism and in which case it is activated. In 1978, academician Emmanuel from the Institute of Chemical Physics, he developed a special classification of aging types. You see how you can prolong a healthy life. You see how the aging process looks like, aging curve, and what happens to the tumor. In the first type of tumor develops at the same time, and you see how depends the incidence of tumors. Academician Yashin, present here, proved a few years ago that now a very unusual situation has emerged. It's not a simple thing. People started living longer. Now, every 10 years, the number of centenaries is doubling in the developed countries. And it seems to me that based on the US statistics, the malignant tumor incidence in the United States is now also going down, even though the death rate is stable. But again, death rate does not depend on external factors. Look at this picture. It was made for mice and for rats. Because in early last century, which means that we live till 70 or 80 years of life, now the first type is there. If we mean to decelerate the rate of pathological aging, we will soon see a situation of successful aging. Not all the scientists believe in it, but let's see. What are the open problems, unsolved problems? The question still is at what age the treatment should be initiated. The majority of experiments show that the earlier you start, the better. The role of sex. We notice that some geroprotectors influence in various ways males and females, and we need to find an explanation for this. Evolution aspects, pathologies, and other problems affect the knowledge as well. In oncology, this is a very good system. There is an international agency for cancer studies in Lyon, in France, which on a regular basis assesses the materiality of proof provided and said to have certain effects. And you know, it's a very deep analysis, calculation based on all the data provided. I think today it's very important to organize a similar system for gerontology, because unfortunately now there is a lot of non-compliance advertising, drugs that do prolong life. Polyphren, which is sued or structured, can increase twofold the lifespan. I'm an expert. I don't trust one result, because of course everything is a start in changes. We need to unify the criteria of assessment of biological age of various species of animals, and we think we have a lot of time to study all of this. And we don't yet have a very clear notion of what geroprotector is, by the way. Because many anti-aging drugs are advertised without having any actual anti-aging effect, or with very little proof that they decelerate the aging process. This is a very important result. In Max Planck Institute in Rostock in Germany in 2003, it was proven that Germany has made a huge experiment, because this and when Germany split into West Germany and East Germany, the death rate in the mortality in the East Germany was much higher than in the West Germany. After Germany got united again, the mortality in the East Germany went down again. And it shows that if we don't do anything about it, we will regret. Because we can do a lot, we can improve the quality of life, and Germany proved it. I think we'll still wait a little bit before we prescribe these drugs. Because the quality of life at an old age is a major factor. A major factor of increasing our lifespan. And if we want to live a long life, we want to be sure that we will not be living until 150 years in a wheelchair or in Alzheimer's hospice. And here you can see my colleagues with whom I've been working for so many years, my colleagues with whom we've made together the majority of experiments I've been talking about today. Thank you. Okay, we have time for some questions. Tell me something. The increased dose of vitamin C, does it increase lifespan? We don't have such proof. But you know, Linus Pauling and so on. Yes, but we don't have any proof of it. Tell me, please, you said that full members of the Academy of Sciences live longer than corresponding members who live longer than doctors of medical science. At the university, we were told to calibrate the device before we start measuring something, and we know that if you want to become an academician, you cannot become it before 60 years of age and so on. So if you adjust the scale, do you think you are right in what you said? You know, I got a month ago an article from Max Planck Institute. The article analyzed the lifespan of academicians of two major academies of science. It's Royal Society of the United Kingdom, which is Academy of Sciences of Great Britain, and the Russian Academy of Sciences, for the whole history of these academies. And it turned out that British academicians, based on the calculations with calibration and so on, they live 3.5 years longer than Russian academicians. Maybe there are some genetic effects we don't know. But you know, people with high income live longer than people with low income. This is a fact, a proven fact. The length of telomeres of people with higher education is greater than that of uneducated people. So how can we calibrate the comparison? There are too many questions we can't answer yet. It's clear that Politburo members of the Central Committee of the Communist Party, they lived much longer. Unless they were shot by Stalin, they lived much longer than average citizens. We know why, because they had very good health care, they had special nutrition, even water they drank was brought from special springs. All of this matters, guys. Any other questions, please? You know, I was intrigued by your data on the athletes. Do I understand that athletes with highest achievements live longer? Because as far as I have heard, that sports reduces lifespan of athletes, that exercise increases lifespan, and active sport reduces it. You know, I also used to think so, but then I looked at the Steramoto meta-analysis of 2010, analyzing 19 sports from many countries, analyzing the lifespan of athletes from national teams. What matters in terms of longevity is the type of sports. We know that members of women's national athletes teams have a higher incidence of breast cancer, higher than in students, for instance. On the other hand, you have to remember whether the sports is a static or an active one, aerobic or anaerobic, like weightlifting and running. You showed a very interesting result about the length of the daytime and the light. You showed that the rodents lived longer, not only when they had 12 hours of light, 12 hours of darkness, but especially they lived much longer when they spent all their life in darkness. What does that mean? Do you think it would be the same for humans? Not really. Not really. The results we have got show that 24 hours of darkness a day does not increase our lifespan. It decreases the risk of development of tumors, while the lifespan is similar to control group lifespan or a little bit less, because it's still a stress, but it has a very good effect on tumorigenicity. Tell me, please, do you have any studies on the lifespan of aviation personnel, because their circadian rhythms are disrupted, but air hostesses, they always look like $1 million. If you see air hostesses 45 years of age, they always look stunning. You know, the results I know of show that the air hostesses have a higher risk of breast cancer, and pilots flying to very faraway destinations have a higher incidence of prostate cancer. That's why they retire at an earlier age. Thank you. Thank you so much, Mr. Anisimov, and thank you for your questions and your answers.